Hey guys, so I guess I missed some information from the previous video. This is the prequel to that video. So let's begin by realizing our topics for this unit. They are Mendelian genetics, Punnett squares, non-Mendelian inheritance, inherited disorders, and pedigrees. And if you're wondering what the case study that we're doing right now for Jenny for my period 3, it is part of inherited disorders. Some vocabulary that we are learning and will be covered through this video and the next video, which you've already seen, are carpal, cross-pollination, dominant, F1 and F2 generation, fertilization, genotype, heredity, hybridization, inheritance, model organism, and monohybrid, P generation, phenotype, recessive, stamen, trait, and true breeding. So let's begin by the very beginning. Before we can understand inheritance, let's understand the process of which gametes can give DNA to their offspring. Okay, so the process begins by fusing two gametes, and that would be the sperm and the egg, and that is called fertilization. The fusing of two gametes is called fertilization, and the genetic variation that we get in our populations created by fertilization process and sexual reproduction. So let's begin by a question that we should be tracking throughout this video. What are the rules of inheritance that Mendel discovered? At the time of Mendel's time, there were two prevailing hypotheses. One is that blending inheritance, and two, inheritance of acquired characteristics. Blending, it basically means what it means. If you take a red flower and cross it with a white flower, you get a pink flower. That was one thought that was going on at the time. Another one, or another competing hypothesis was that if you want a specific trait badly enough, you will get it. And the example of that was a giraffe extending the long neck because its ancestors kept reaching for these leaves on the trees, so eventually all of their offsprings got that trait passed on. So Mendel was trying to disprove one or the other. He started out by thinking about the organism that he wants to test this on. And a model organism has to have specific characteristics. It has to be small, short-lived, inexpensive to take care of, produces many offspring, and can be easily manipulated. What better plant other than that pea plant that he thought about? Mendel had a lot of time on his hands. He started out by controlled mating. He wanted to make sure that he mated the same parent generation for a little while before he can experiment and cross them together. Each pea plant has both reproductive organs. It has the stamen, which is the pollen producing, and it has the carpel, which is the ovule producing organ. The stamen is the equivalent of a sperm. The carpel is the equivalent of the egg. So that was controlled because he could basically cut off some of that stamen before it matured and put it on the carpels of another organism if he needed, or even the same one if you wanted to either cross the two together or just do a controlled breeding on one. Controlled breeding, as you can see over here, he used the scissors and I believe a paintbrush, and what he did was he cut off the stamen of the plant to ensure no unwanted sperm would touch the carpel, and then he would use a paintbrush to apply that specific sperm to the carpel. Okay? So Mendel's phenotypes. The peas are not only the color of the flower that he observed. He observed the form, the cotyledons, the color of the flower, the pod form, the color, the stem place and size, and altogether gave him 7 phenotypes. So all of these, each one of these is a phenotype, and for each phenotype there's two characteristic traits, either yellow or green, white or purple, puffed or full. The place of the flower could be in the middle or on the edge. The size could be short or long. So those were 7 phenotypes, and we know that phenotypes mean traits. 2 of our vocabulary words that are in our list. Okay. Oops, did not do that. Now, typical experiment. True breeding was the first part of Mendel's experimentation. Before he can cross anybody, he wanted to make sure that he got the same phenotype over and over and over and over, so he can make sure that that pea stays yellow no matter what and is a true breed. So that is called self-pollination. You self-pollinate a flower with itself and you get the same exact flower again. Eventually, he would do hybridization. And basically that means taking 2 true breeding individuals and crossbreeding them together. Hybridize. So these 2 are 2 true breeding plants. This one is a true green pea plant and this one is a yellow breed pea plant. Crosses them together and we get the F1 generation, which is all yellow. And then we cross these 2 again with each other and we get, wait for it, yellow and green. And I think I I began my last video with monohybrid cross, but what it means is that you are analyzing one trait. So in this cross, we are taking care of or observing the flower color. So as you can see here, we got 1 purple and 1 white for the parent generation, and then for the F1 generation, we got them all purple. And this is where I end. So I'm going to end this one. Or actually, let's make the connection to the monohybrid cross of the second slide. If we have 2 F1 generation purple flowers crossed with each other, we will get a ratio of 3 to 1 purple to white, or 75% purple and 25% white. Now, is this a pattern of inheritance? And I believe this is where I left off my last video, so I'm going to end this one over here.